The broad concept of this paper is surrounding topographic representation in the human brain. According to this organizational principle, groups of neighboring neurons encode functional properties of the same specific body part. The size of the cluster devoted to said body part is proportional to its functional importance. This seismic organization is illustrated in what is called a homunculus, where the drawn size of body parts is proportional to their use. Generally, the hands and face have the largest representation. Now, it is widely known that the sensory and motor cortices are represented topographically. However, in this study, the experimenters investigated whether or not this same topographic mapping of the human body exists in the occipital temporal cortex.
The occipital temporal cortex, which we will refer to as the OTC, is located around the junction of the occipital and temporal lobes. As a higher-order visual area, the OTC is known to be involved with perception and specifically object recognition. Past studies have further identified distinct areas in the OTC to be activated by facial and body images. These areas have been defined as the extrastriate body area, or EBA, fusiform body area, FBA, occipital face area, OFA, and fusiform face area, FFA, located in the designated green star region. In order to assess OTC activation corresponding to different body parts, the researchers conducted a phase encoding experiment on 12 participants. This involved voxel-wise analysis across the OTC. A predictor function based on each voxel's raw bold signal was time-shifted to match each voxel time course with the presentation of the body part that produced the highest activation. This was necessary due to the hemodynamic lag of the bold response. Using this data, the experimenters developed body part activation maps for each individual participant, and then performed group averaging to generate an overall group map. The body part categories presented to the participants were as follows: upper face, lower face and neck, upper limbs, and trunk and lower limbs. The body parts were viewed in a double wedge, rotating around a central fixation, which was completed a cycle in 48 seconds, as seen at the top in Figure 1a. Each body part category was presented sequentially in a fixed order, and each cycle was completed in 30 seconds. To control for ordering effects, the experimental design was repeated for 3 or 4 runs with a reversed or shuffled body part sequence. Figure A shows the hemodynamic convolved response function that has been shifted along a run to determine the highest R value for a given voxel. This maximal R value occurred when the voxel's preferred body part was presented. Figure B shows 5 voxels and their corresponding activations to body parts for 3 runs, and then the last column being their average activations. Voxels with a maximal R value above a determined threshold were then color-coded based on its preferred body part and its location shown in Figure C. As you can see, similar body part preference appears in clusters of neighboring voxels. Figure D shows the strength of body part preference. This was determined by subtracting the R values from the most preferred category from the second best category. Green areas indicate high difference between the R values, while red areas indicate low difference between the R values. The red areas were found near the borders of the clusters, suggesting that there was a smooth transition between body part selectivity for neighboring clusters. The researchers averaged the voxel R values for each of the individual body maps using Tellerac coordinates. A consistent topographic organization is seen at the group level in this image as well, with distinct clusters showing preference for a particular body part. Representations of the upper face in green lay in the well-described FFA and OFA bilaterally. Distinct from the upper face, the lower face clusters shown in blue were located bilaterally in the OFA and the right FFA. The upper limbs were located in the dorsolateral section of the OTC, indicated by the yellow area. Both the lower limb and trunk representations, pink and red, respectively, were distributed between the dorsal and ventral aspects of the lateral OTC. It's worth noting that the body maps extend beyond the boundaries of the EBA and other areas previously defined in studies. Through the phase encoding experiment, the researchers were able to define regions of interest for each of the 5 body parts. They then conducted an independent block design to compare activations of a centrally presented image for each body part, as well as a scrambled version used for baseline. The results further confirm that these specific regions showed the same body part preference as defined in the face encoding experiment. The bar graphs in Figure B represent the cross-correlational R-values of voxels within the ROIs for each body part. The R-values were determined in the phase encoding experiment. The bar graphs in Figure A show the beta values, or mean activation levels, for each body part within the ROI determined by the block design experiment. As you can see, preference for each body part was replicated across both experiments for each of the ROIs.
To determine if body part preferences exhibited in the previous experiments were simply due to stimulus shape, the 5 categories were further divided into subcategories. The upper face was divvied into eyes and nose, the lower face became mouth and the chin-neck area, and so on. The activation levels induced by each subcategory were tested in all of the defined regions of interest, and correlation between each related subcategory was also analyzed. In this figure, the bar graph represents group correlational analysis, where the mean R values were between 0.67 and 0.93 with less than 1% room for error. The scatter plots take the same evaluation down to the individual level. And as can be seen, the highest activation occurred when viewing the 2 subcategory parts that comprised a given region of interest's original preferred body part preference. This illustrates that shape alone cannot explain the specificity for body parts. It is possible that the body map incorporates body part-specific motor information. To test this, a subset of the 10 original participants performed unseen movements with their tongues, hands, buttocks, and feet in a block design fashion. Using the general linear model approach, beta values, aka mean activation values, were extracted from the visual and motor block experiments for each body part from each participant. The figure shows bar graphs representing the visual and motor block activation results, and the scatter plots depict the correlation between these 2 conditions. The 2 measures were significantly correlated in the upper limb, trunk, and lower limb categories. Mean R values were between 0.73 and 0.93. Interestingly enough, the only region of interest that lacked a motor selective representation was the lower face and neck, possibly because we rarely observe movements here in ourselves or others. These results illustrate that it's possible that the body map in the OTC has a role in visual monitoring of movements. It is possible that this visual-motor relationship is due to mental imagery evoked by motor execution. 2 groups of participants either imagine themselves viewing movements executed by a body part, or they imagine themselves performing movements with this body part. The slight activation produced was not significant enough to elicit a topographic representation of the body in the OTC, confirming that the motor-related OTC activity isn't due to the mental imagery or visualization of body parts.
In their discussion, the researchers identified 5 different organizing principles that could dictate the body part organization seen in the OTC. First and foremost, the nearest neighbor similarity principle, where neighboring neurons share this similar preference, is a fundamental principle in understanding cortical map topography. This was shown in the study as neighboring voxels had similar body part preference. Shape similarity, though shown not to be alone sufficient in generating body maps in the OTC, may still play a factor and contribute to the organization of body maps at a higher resolution than depicted in this study. The eccentricity bias principle, which constitutes preference based on the tendency of a body part to be in the center or periphery of the visual field. For example, faces appear more centrally in the visual field as they are foveal representations, while limbs usually appear more in the periphery. This is seen to some extent in the study, with the face being topographically closer to fovea-based representations in retinotopic mapping, while the limbs and trunk were more peripherally based.
Within the body map, physically adjacent body parts were often mapped onto neighboring clusters, i.e. lower face was between upper face and trunk. Non-preferred body part categories elicited weaker activation in regions of interest as their physical distance from the preferred category increased. This was only violated in the upper limb region of interest, where the second best category was lower limbs rather than the trunk, possibly due to their similar shape or motion-related characteristics. In this experiment, static images were presented, but it's possible that some shared representation between the limbs and the upper limb region of interest is due to implicit association between their similar capacity for motion.
The researchers were able to present evidence that higher-order visual areas also represent information in an orderly map and provide categorical information regarding the body. They also investigated and found evidence supporting a relationship between visual and motor inputs with respect to body part representation in the OTC. This provides an interesting area for further study. Furthermore, they were able to show that imagery of body parts and also shape similarities between body parts were not alone able to produce body maps.
The experiments carried out in this paper uncovered several plausible theories that require more research to be either confirmed or denied. Firstly, further inspection of visual motor relationships in visible versus non-visible body parts could be very useful. This could provide further understanding as to why there was no significant correlation between these two epochs in the lower face and neck region of interest. Secondly, an experiment that adjusts top-down body part-specific attention during a motor task may enhance the level of activation in visual representations of that body part. This could provide clues about visual motor learning and imitation. And finally, it is unclear whether the visual motor correlation of preferred body part categories occurs at the individual neuron level or whether there are two distinct and modality-specific neural populations selective to the same body parts. A repetition suppression experiment could clarify this.